कर रहे फिर यूट्यूब पे ये डालना चैनल का नाम उसका IARANHP कास्ट NHP कास्ट
So, good morning friends. Today we have a very distinguished
हो गया भाई कर दिया मैंने
So this is the first time we are experimenting
waiting for
the option of linking universities in the lecture. For the last 3-4 days we are facing one of the issues. Yesterday it went on but there were no issues. Right. So shared na? Thing is working na?
Yeah.
Waiting for confirmation. So the idea is in future, so no one will be invited here, right? You sit in your college or universities, wherever you are, lectures will be delivered from you. So to start with, we already drafted a course. It's the 10 sittings. Each sitting is for about 25 to 30 minutes. That's what the education is saying. You cannot engage students more than 30 minutes if you want to convey something effective. Right. The topic we have identified is molecular basis of post-conjugate interaction. So it is only the entire course is condensed in 10 sittings. Each sitting is 25 minutes and 5 minutes discussion. So, and each sitting, the number of slides are not more than 25 slides. So this is now going on. I think the modalities have already started, and maybe in a month or so we will be launching that one. So we will be inviting university students to register for that course. It's the same mechanism. You have to register online, and it will be verified here, and they will be given a kind of certificate once you have done this course. So whatever happening now is a small step towards that. Got the link? Sure, it is not connecting. Link is not connecting.
Sir, it's working. We are live.
But it is working here. Can you check? Can you confirm once it is linked? I'm waiting for that. Okay. Or shortly it will be something like that. We have not yet started. What is the screen display? Okay, I'll check it. Good morning. It is getting connected? Yeah. It is connected now. Right, okay. Okay, thank you. Yeah. It's getting.
So, good morning, friends. Sitting in our ACTP auditorium, and at the same time I also welcome student from US Bangalore and student of AC and RA Kilikulam. So today we have a very distinguished speaker with us, Dr. A. Kumar, who is having about 28 years of research experience in the field of plant pathology with a special training on microbial genomics. He has handled several projects on associated microbes and mitigation of bacterial and fungal oomycete diseases of crop, especially on black pepper, ginger and rice. And he has worked on various biocontrol pathogens, especially this DBT. He has been a DBT overseas fellow at Wageningen University, Netherlands. Dr. Kumar worked on molecular aspect of Pseudomonas aeruginosa, strain BP35 and Indophyte, and Pseudomonas fluorescens SS101 and SBW25 for rhizobacteria under the supervision of Dr. Josh Rajmakers, an authority on Pseudomonas based biocontrol system. He is very well conversant with the several modern molecular techniques like genomics, bioinformatics, tool related to genetic transformation of bacteria and fungi, structural and functional analysis of microbiome and microbial gene encoding, antimicrobial metabolites and surfactants, antibiotics. Recently, he has completed a project funded by DBT, Government of India, which deals with the genome mining of endophytic bacteria and for identification of novel biofunctional compounds and their biosynthetic genes of crop protection. His other scientific interests are in the area of non-host resistance in rice against biotrope, use of genomic tools or to understand plant microbe interaction, whole genome sequencing of plant pathogens, plant microbiome, and use of microbial elicitors for sustainable disease management in crop plants. And he has published more than 125 publications in leading international and national journals, and he has guided more than 22 PG and 5 PhD students. Presently, he is playing mentorship to 1 MSc and 4 PhD students in the division, and he has completed about 12 externally funded projects sponsored by DBT. So with this brief introduction, I request Dr. Kumar, please start your lecture.
Thank you, Dr. Pant, for your kind introduction. Friends, sitting here, I'm sitting under the two universities. This is a very basic lecture on real-time PCR. And basically, I brought this presentation to make you understand the fundamentals of this real-time PCR technology, which is one of the most important technology in plant pathology and plant virology in particular, to understand the biological questions each one of you is addressing in your thesis project. I'll get into the presentation per se. If you have any questions, let us reserve it for the end of the presentation. And additionally, you can send the questions to my email as well. This lecture, basically, I would like to cover certain aspects like very brief history of this technology, that is real-time PCR technology, and the terminologies and acronyms. Many times you encounter different acronyms in real-time PCR technology. We take a snapshot of what they really mean to us in one slide. And the history, of course, I highlighted that. The major differences between PCR, that is a conventional PCR, and the real-time PCR, or PCR, would I put it there. And of course, the technologies, what is being used in real-time PCR technology, what is the dye-based qPCR and also the probe-based qPCR. Here comes, I would like to have your attention. What is the fundamental difference between these two independent technologies that is currently used in real-time PCR? And some basics about, different kind of jargons people use in real-time PCR technology, FRET, non-FRET kind of quenching. We will have a couple of slides on that, and we'll get into the analysis of real-time PCR, how the different analytical tools are being used in real-time PCR. One is the absolute quantitation and also the relative quantitation. Most of you may be doing or may not be doing, but you will take this opportunity to fine-tune your knowledge on this real-time PCR. And of course, the applications many of you may be knowing. Let us take some questions on that, and let's have some interactions as well. So, the technology, that is real-time PCR technology, is also called as a qPCR technology. That is nothing but quantitative polymerase chain reaction. Right? It is a method by which the amount of PCR product, PCR product, you understand what is PCR product, also called as amplicons. It can be determined in a real time. So the title or the name itself is suggesting that in a real-time basis, you can monitor polymerase chain reaction. It is as simple as that, right? And it is often abbreviated as qPCR, and this method is also referred to as many times real-time PCR. So here comes the two names for the same technology. One is real-time PCR, another one is qPCR. So one is indicating that we can monitor the reaction in a real-time basis, hence the name real-time PCR. And the benefit of this technology is you can quantitate how much of nucleic acid is there. So the name Q, that is quantitative PCR. So these two, this is synonym, there is no differences also. And one of the biggest advantage of this real-time PCR technology is it does not rely on any downstream analysis to visualize the results of your PCR. What I mean to say here is, if you think of your normal PCR, if you want to know the results of your normal PCR or conventional PCR, you need to have a separate options of electrophoresis or dense temperature in this particular case to visualize the results. So in this particular qPCR, you don't need that because you can monitor the progress of polymerase chain reaction in the real-time basis, right? So it is extremely versatile, enabling multiple PCR targets can be assessed simultaneously. This is another big advantage of real-time PCR. You can amplify multiple targets parallelly at a time. So this is the third advantage added here, right? Number one is you can monitor the reaction in a real-time basis. Number two, you can quantitate how much is getting amplified. And number third advantage is multiplexing is possible in a single reaction prediction. Very useful for an application like gene expression. Most of you must have heard about it. When the virus is infecting a plant, and what is really happening in the plant metabolism, if something is happening in plant metabolism, it goes down up to, or it originates from the so-called transcriptional changes in the host upon virus infection. So what is getting expressed and what is getting depressed, all those things can be analyzed using real-time PCR. The key feature in real-time PCR is we will be focusing more on that only. And in fact, the half of my presentation is centered around the tools and probes and the tools and dyes, what is being used in real-time PCR. Let's see what has really happened in the past that eventually led to invention of a tool called real-time PCR, right? This is a timeline of events. It happened at different time points in the history of genomics. And the tipping point happened in the year 1985, right? So in 1970s, people gave a concept like DNA can be synthesized or rather replicated outside the cell system in a small tube. So that was a concept, right? Maybe it was a very revolutionary concept in those time points. So at different time points, you just read the literature about in vitro synthesis of DNA. So that was the original concept of the PCR. So different timeline, gradually information started coming in literature, and in 1983, the original concept for replication of DNA outside the cell system in a small tube was demonstrated. So that paved the way for the so-called machine, the thermal cycler. You may call it as polymerase chain reaction PCR machine. So that started coming to the market from 1985 onwards. And in the year 1989, one of the key ingredient in PCR, that is the enzyme which is doing the polymerase function, and the journal Science has rated this molecule as the molecule of the year. The title was according to this particular design, right? In 1993, the inventor of this particular technology was awarded the most prestigious award in science, that is Nobel Prize. So the nucleic acid amplification and the detection techniques are the most valuable tools in biological research. All of you are sitting here, especially those who are working on viruses. They are the starting point. Whoever is dealing with viruses, in fact, you need to amplify the so-called viral nucleic acid in order to study them in vitro. So it is integrated in biology, in particular the plant biology. So you cannot do away these techniques if you want to really know more about plant viruses, right? And every branch of science, leave alone pathology and virology, in every branch of biology, you will find the role or the key role played by PCR. You cannot think of biology research nowadays without polymerase interaction. So we have reached such a stage. I'm sure whoever is working on any aspects of biology, if you want to invent something new or if you want to find something novel, you need these technologies, right? You cannot ignore this technology, be it agriculture or biotechnology or medicine, forensic sciences, diagnostics, plant diagnostics or human diagnostics, animal diagnostics. This method is very used in a very prolific manner. And in those days, before the invention of this real-time PCR, people were using this normal conventional PCR for various applications. In those applications, we will be seeing whether something is getting amplified or not. So that itself is good enough to say whether the target is present in the sample or not. So later on, the scientists felt that it is not what is getting amplified alone is sufficient in biology, but we also need to know how much is getting amplified or how much is really present in the original sample. So here comes another requirement in biology, what is called as the quantitative terms. So conventional PCR and real-time PCR, the fundamental difference is in conventional PCR, you are bothered about what is getting amplified. But in the case of real-time PCR, you are really bothered about how much it is getting amplified. That's fundamental difference, right? In real-time PCR, can be used for both, of course, for both quantitative and also qualitative, because while pursuing that quantitative analysis, you will also come to know what is really getting amplified. And that's advantage. So in other terms, I would say that this real-time PCR technology gradually is what they call making the conventional PCR obsolete. So that's what is going to happen. It's already happening. So very soon, the real-time PCR technology will be one of the most important one. Anybody? Who is this person displayed on the screen? Yeah, he is a person who invented PCR. That was in 1983-85 period, and unfortunately, he passed away very recently. That was in August 7, 2008. So it was big news in among the scientific fraternity. So Carrie Banks Mullis is an American biochemist. In recognition of his invention of the polymerase chain reaction, that is a PCR technique, he shared 1993 Nobel Prize in Chemistry with another scientist, Michael Smith, right? He also contributed enormously on nucleotides and nucleotide chemistry, Michael Smith. So what happened most the parallel time, 1983 to 93, the 10 years time was the period people started improving the technology of in vitro synthesis of DNA. So a lot of innovations happened in a rapid manner. So 83, the concept was proven in the lab. 85, the machines started coming to the market, and 93, Nobel Prize was given. Parallelly, another group of scientists who was working for Roche Molecular Systems Incorporated, they started thinking in a different way. That the thinking process has culminated in the concept of real-time PCR. So what I mean to say here is, in 10 years' time, the technology is completely taken a new turn in science. In conventional PCR, it is blossomed into a new technology, which is called as real-time PCR. That has happened in 1993. So let's see what has really happened. This is a Nobel Prize in chemistry in 1993. I just brought this picture from Nobel Prize, the original website.
And this is a statement what is given there. So the Nobel Prize in Chemistry 1993 was awarded for contributions to the development of method within DNA-based chemistry, jointly with the one half to carry the Willis for his invention of polymerase reaction method, and with one half to Michael Smith for his fundamental contributions to the establishment of polygonucleotide-based direct site-directed metagenesis and the development for protein studies. So, 1993 Nobel Prize was shared by two. We only remember Gary Willis, but nobody remembers Michael Smith. So at least you carry his name also in your memory now. So equal contributions, right? So yeah, this is a paper. If you want to get into the original paper, which brought out a new concept, maybe he was not aware at that time. This is a technology which is going to change the biology once for all. So that was really a landmark achievement or invention. What do you call that? The kinetic PCR analysis, the real-time monitoring of DNA amplification reactions. This concept emerged in the labs of Roche. They improvised the PCR and they came out with the concept, the kinetic PCR analysis. This original paper appeared in Nature Biotechnology. And parallelly, there is another small journal, a prelude to this paper that appeared in Biotechnology, volume 10 in 1992. Simultaneous amplification and detection of specific DNA sequences. That was the original concept, which was proposed by Higuchi. Higuchi, at that time, he was part of the Roche Molecular Systems Incorporated team, and they were trying to improve the conventional PCR into a real-time PCR, and they brought out these two publications in a quick succession in 1992 and 1993. And these two publications are the preliminary publications which eventually led to the present-day qPCR technology, what we are doing in science now, right? This is in a nutshell about the history of real-time PCR. In 1993, Higuchi et al. Incidentally, in that same year, the Nobel Prize was given to PCR technology. So in the same year, in 1993, he recognized the process of PCR could be monitored. So this is a small intervention. So PCR is already there, and we really don't know what is really going on the moment you shut down the machine and you close the door and put on the machine, whether the PCR is really happening or not. You will not be able to know until you spend another 3 hours in running the sample in gel electrophoresis, and after maybe from the start to end, maybe 6 to 7 hours of time period, you will come to know whether the PCR was PCR has happened or not. So that was the what do you call the time to spend. But here he came out with the concept right from the stage you put on the machine. You can monitor what's really going on in the small tube where the cocktails of PCR is already mixed. So the concept here was he added extra ingredient in the PCR machine, and with the help of the extra ingredient, he made the system what do you call seen to the machine and also to you through a small chemical intervention. So that is what is called as fluorescent label that binds to accumulating PCR product. So remember this point. So a conventional PCR, and it is added with fluorescent dye, and that fluorescent dye has got a very high affinity towards the so-called amplicons. Amplicon means it's a double-stranded DNA, and once it is bound with the double-stranded DNA, and they brought a kind of an extra attachment to the PCR machine so that the PCR reaction can be monitored. So that's what exactly happened in this particular concept. So if you see the original papers, they used the dye, what is called as ethidium bromide. I think all of you must be knowing. This ethidium bromide is intercalate to the DNA, and that makes the product visible to the machine. Voice is audible?
Yeah.
Live stream it is not audible. I think I'll speak with closer. Yeah, I'm sorry for that. I think hope it is audible hereafter. As the concentration of PCR product increases, the intensity of the fluorescent signal also increases,
and in those times they have used a chemical what is called as ethylene bromide. That is for the detection. So the discovery really paved the way for the modern day quantitative real-time PCR. The only difference is people stopped using this ethylene bromide and they replaced it with much safer dye in real-time PCR, and those dye we'll be having some discussion. And in 1996, another technology came. They used a new concept of using a Tachman detection method. We'll be having a detailed discussion on that. And in 1996, much similar to the invention of PCR, and the PCR machine came in 1985. Here too, in 1996, the first machine which can do this real-time PCR came to market. That is from RBI, it is Applied Biosystems. They introduced first real-time PCR technique. This is sequence detection system. That was ABI 7700, that was the machine. So you can see this is the first real-time PCR machine ever came to market. And this machine helps you to set up real-time PCR in your laboratory. So little bit of the acronyms. Acronyms, what is used in real-time PCR. Of course, qPCR, RT-PCR, and qRT-PCR. I think this terminology is quite familiar to all of you, right? What is PCR? That is nothing but polymerase chain reaction. qPCR is a quantitative PCR, which I already explained. Real-time PCR, RT-PCR, sometimes it is confusing. People call it as a real-time PCR. And also we have another expansion for that, that is reverse transcriptase PCR, right? And we combine these two, we have another one, qRT-PCR, right? So the question here is which one to use in your day-to-day research activities or in your publications, right? So now it is accepted by many, if not all. The term qPCR is the most accepted one that conveys somebody has used real-time PCR in their experiment. So I would prefer to use qPCR to make it distinct from the conventional PCR. So it also conveys that somebody has used real-time PCR, right? This is, the acronyms are well integrated in biology now, but if you see literature, all these acronyms are very frequently used by different people, but I would prefer the qPCR as a standard one so that it no need to confuse the readers of that particular paper. Now let's come to the finer details of these differences in conventional PCR, which I highlighted already. What is amplified is the focal point of conventional PCR, and how much is amplified is the focal point of real-time PCR, right? Is it audible now? You can find out. Now also it is not audible. So what to do, Prashok? Yeah, but in other universities it is not audible. Okay. Yeah, it's really audible here. That's what the technician informs me. Perhaps you can check the audio system in your university, and you can find out from maybe the Agriculture College and Research Institute curriculum whether it is audible or not. If it is audible there, then perhaps the problem is with the other university. I think the no audible, it is from US Bangalore. Yeah, I think let them check. You can find out from maybe curriculum whether it is audible or not. So that would resolve the problem. Is I think it looks like the problem is not from here. Let me continue this, right? Yeah, what is amplified is the focal point of the conventional PCR, and how much is amplified is the focal point of real-time PCR, right? This is a specialized technique which allows you to monitor the polymerase chain reaction in the real time, right? Yeah, I already highlighted these points just to have a recap of the technique. You can go through it. Quantitative real-time PCR, a method of polymerase chain reaction that allows to follow in real time the amplification of a target, hence the name real-time. The real-time PCR technique is a technique in which a probe bind to a specific target region of the amplicon to produce
fluorescence during PCR. So it is nothing but a conventional PCR added with one extra ingredient. That extra ingredient is fluoroprobes. Okay, you're trying being you reserve this, understand this. The fluorescence measured in real time is detected in PCR cycler with an inbuilt filter fluorometer. So if something is extra added to the PCR mixture, then the question is, what is the benefit of adding this chemical to the PCR mixture? In what way it is going to help us to monitor the reaction? So for that, you need to have an extra attachment, which I already highlighted. So in other terms, two interventions here at the time of PCR master mix preparation and also at the time of setting up the reaction, there is some modifications. In the PCR master mix preparation, extra ingredient is added. Hence, there is a change in the routine composition of PCR, number one. Number two, the machine is also completely transformed into a different machine where the PCR, normal PCR machine is added with an attachment. That attachment is able to monitor what is going on in the small tube, right? So here comes the pictorial depiction of the real-time PCR machine, what is really inside. So in a simpler language, real-time PCR machine is a PCR machine combined with a monitoring system. The monitoring system here is called as spectrofluorometer. Try to understand this. A PCR machine which is attached with an extra attachment. That extra attachment is called spectrofluorometer. So the spectrofluorometer, which monitors the reaction, what is going on in PCR tube, right? Let's see how it really happens. This is a spectrofluorometer, which is kept on the top of the PCR machine, where it can oversee what is going on in the tube, right? In the bottom, what you have is the so-called PCR machine. So in other terms, a PCR machine, which is attached with a spectrofluorometer, it converts a normal PCR machine into a real-time PCR machine in a simpler language, right? And here comes the light path. Always remember, there is one excitation wavelength, another one is emission wavelength. I hope you understand the difference between these two, and these two are very important in real-time PCR, right? And it emits a specific wavelength, excited by a specific wavelength. So let us get into a little more details. So this is a typical conventional PCR. We have anybody, how many ingredients are there in a typical PCR reaction? We have the buffer system. We have the so-called the substrate, that is a DNTPs. We have some cofactors, that is magnesium we normally use, right? And we have primers which prime the amplification, right? And you have the template DNA, and last but not the least one, so very, very important, that is polymerase chain reaction. So this is a conventional PCR system. So a PCR reaction. So if you add that extra chemical, that extra chemical is also called generically a detector. So what it detects? It detects the amplification product, right? So the detector addition would make a conventional PCR into real-time PCR. This is a detector. And if you see the literature, there are two different kinds of detector, very, very popular in real-time PCR. So the first one is the so-called dye-based detector or dye detector, also called as a non-specific detection. We'll have a detailed discussion why it is called non-specific detection. And another one is specific detection, what is also called as detective probes, right? Try to understand this. One is a fluorescent dye, another one is detective probes. These two are parallel technologies which is used to monitor the PCR reactions, right? And this technology, the dye-based one, is also called as dye-based quantitative real-time PCR, and or dye-based quantitative PCR, and another one is probe-based quantitative PCR. Let's take one by one. Dye-based quantitative PCR, right? So we know that ethylene bromide was the first one used in real-time PCR, right? Maybe the original inventor. So later on it was replaced by the safer dyes in the later years, and the dye what is called as cyber green. If you do not know, please remember this name. It's a dye which is used in real-time PCR reactions, and this dye intercalate into double-stranded DNA, right? And it has got a very specific affinity towards the minor groove of double-stranded DNA, right? If you try to remember this point, and very specifically binds to the minor groove, right? So once it is bound to the minor groove of the double-stranded DNA, the fluorescence intensity of this dye is increased, right? So of course this has got a small disadvantage, that is it can go and bind to any double-stranded DNA minor groove. There is absolutely no specificity whatsoever. Whatever double-stranded DNA comes in its path, it is going to bind with the minor groove, right? For that reason only this is also called as non-specific detection method, right? Remember, all cyber green based detection is called as non-specific detection method, right? Yeah, cyber green is all alone. We have several options are there, and those options are the listed here. I'll just take a stop here. Whether the audio is still working there? Okay. Okay, they have not confirmed. Okay, let us believe that it is working. Or you can send a query. Huh? Yeah, you send a message, yeah. Okay, let me continue. So it's not cyber green alone, which can be used in real-time PCR. There are a number of options are there that is listed here. Cyber green 1 and 2. You have Eva green, LC green, Bibo, YoPro, and Cyto family. These are all some of the options. But the most popular one is cyber green, right? So one of the criteria for selection of most appropriate dye in real-time PCR, it must be thermostable. Number one, because in PCR, you are subjecting the cocktail to very high temperature of around 94 to 96 degrees Celsius. So the dye must be tolerant to very high temperature. That is one of the criteria. And second one is it is resistant to the different inhibitors, because remember, PCR is all about interplay among different chemicals. So maybe some potential inhibitors are there in the PCR cocktail, and the dye should be completely resistant to those inhibitors, right? These two are the guiding selection, what do you call, for this, the most appropriate dye used in real-time PCR, right? This is a little more on what is this cyber green. So it is, remember, cyber green is a kind of brand name. It's not a, what do you call, generic name. The original name is written here. If possible, try to memorize that, right? So what is written here is NN-dimethyl, and it goes and goes and goes, right? This is actual name of cyber green, right? And the formula and the molecular mass and the finer details are. One point I would like to make a mention here. This is not really soluble in water. It is soluble in a special solvent, what is called dimethyl sulfoxide, right? This is a little more on the cyber green, right? This is the kind of molecular structure, how it, yeah, remember the cyber green family of dye is produced by one company. There is a molecular probes. It is a subsidiary of Life Technology Corporation. I think they are the original inventor of this particular dye for real-time PCR. Let's see how this cyber green, not at all audible. Prashuk, I think there is some serious problem there and there is no audio. I'm sorry for those students who is sitting there and Let me continue now. There was a small interruption because some audio issues in UAS Bangalore. Hope it is getting resolved soon. Let's get into the finer details of the cybergreen, how exactly it works, right? So I made a mention here, cybergreen has got very specific affinity towards a minor groove of double-stranded DNA. I hope you understand what is minor groove, right? In DNA helix, there is a major groove and also a minor groove. You please recall those days. So those finer details, and this has got a very specific affinity towards the minor groove. So what happens at the start of your PCR, you have everything floating in the, in a PCR mix. Your cybergreen is also one among them, right? The moment the PCR is about to start or it's already started, that what happens there. So you please recall how the PCR is really happening. So the double-stranded template will become a 2 single-stranded, and the primer will go and bind to the complementary sequences. Then your polymerase chain reaction, polymerase enzyme will go and extend the primer beyond where it found, correct? Then the extension happens, right? Then what do you call a single-stranded DNA is converted into a double-stranded DNA. So here comes the role of cybergreen. So the moment double-stranded DNA is formed, you can see here the cybergreen molecule will go and bind to the minor groove, right? So in a nutshell, what happens in a solution, the unbound dye exhibit very little fluorescence, right? And the cybergreen, one binds to the minor groove of the DNA double helix. After annealing of the primer, the dye molecule can bind to the double-stranded DNA, in particular the minor groove. The DNA binding results in a dramatic increase in the cybergreen 1 molecule to emit the light upon excitation. So what happens is with every passing cycle, there is an accumulation of double-stranded DNA. So when there is a double-stranded DNA accumulation happens, let us not forget the minor groove is already bound with the cybergreen. So every passing cycle, there is a increase in the fluorescent signal which is coming from the double-stranded DNA. So that means you start seeing the signal in the machine when a sufficient quantities of double-stranded DNA molecules are generated in the form of amplicons, right? This is pictorially, it is given something like this, the progressive increase in the fluorescence as the amplicon accumulates. And intensity of fluorescence is measured by spectrophlorometer, right? So what happens here? Initially, the number of double-stranded molecules are very, very small. So you just think of the mathematics in the so-called DNA amplification, right? Initially, the first cycle, what happens? How many new strands are produced? 2. The 2 becomes 4, right? 4 becomes. So there is a geometric increase in the number of double-stranded molecule, right? So what happens here actually? So the free unbound dyes are getting accumulated into the double-stranded DNA, right? So the intensity is measured by spectrophlorometer. One question I would like to ask here, right? When do you really see the fluorescence in the machine? Anybody has done real-time PCR before? Please raise your hand. Raise your hand. How many of you have really done it? Yeah. So when do you start seeing the indications of the fluorescent signal coming from the PCR mixture? After at least half of your cycle is completed. What I mean, if the number of cycles are around 30, the moment you start crossing around 15, 16, 17, and you start seeing the curve raising up, right? So why it is happening? We will have a separate slides on that. That would explain why there is a delay of around 15 to 18 cycles before you start seeing the fluorescent curve raising up. Okay, that's a very important point. We'll have a small discussion on that. So before I move on to the next slide, some advantages of dye-based real-time PCR. It's very easy. All you need to do is you just add cybergreen in the PCR mixture. If you have a real-time PCR machine in your lab, you are ready for real-time PCR experiments. It's very easy. No complications, right? It's cheap, easy to handle, very sensitive and effective. The disadvantage, which I already told you, it is completely non-specific. So the dye can go and bind any double-stranded DNA, not necessarily it is coming from the target. So keep this point in your mind. Is it clear? That's a dye-based qPCR. So initially you have a lot of free unbound dye in the mixture. After the amplification, the unbound dye is bound with a double-stranded DNA, and the double-stranded DNA emits the fluorescent signal. So that's what happens with dye-based real-time PCR. Let's move on to the second what you call parallel technology in real-time PCR, that is probe-based quantitative real-time PCR, right? So to understand this, you need to have little base information about the polymerase chain reaction and also exonuclease activity of the same polymerase reaction. I think some speakers already made a point here in the same hall. Let's get into the finer details. The probe-based qPCR, it relies on the sequence-specific detection of desired PCR product, right? Unlike cybergreen-based qPCR method, which detect all double-stranded DNA, the probe-based qPCR utilizes a fluorescently labeled target-specific probe, resulting in increased specificity and sensitivity. Hope you don't understand this. The point here is, in probe-based detection, remember, you need to have a probe in the PCR mixture. So I asked one question, what do you mean by probe? And how do you differentiate probe from the primers, right? We all know that there is a primer in real-time PCR, correct? No doubts on that. But in probe-based quantitative real-time PCR, in addition to the primers, you also have another primer-like thing. That primer-like thing, we call them as probe. Very simple. In other terms, in a simple language, the detection is enabled by two different probes. One is called as a primer, another one is called as fluorescently labeled probe, right? Remember, these two molecules, they are a single standard, they are the primers only. One we call it as a primer, another one we call is probe, because that particular nucleotide stretch is fluorescently labeled. Is it clear to all of you? That means what? Before you start using probe-based quantitative real-time PCR, you need to know the target, number one, and you need to have the primer designed for the target, amplification, and also you need to have the probe designed from the same target, right? In other terms, you need to design two sets of primers. One you would be employing or deploying for amplification. Another one you would be employing to bind to the target very specifically, right? And that particular nucleotide sequence is fluorescently labeled, right? Unlike cybergreen, I already told you, it utilizes a target-specific probes. The probe is nothing but the same stretch of nucleotide sequence, different from the primer, but targeting the same nucleotide targets in the genome. Is it clear? If anybody is not clear, please clarify, raise your hand. You have a target, say any gene, right? You want to see the expression of that gene very specifically. So you need to design a primer for the gene. The same way, you also design a region in the genome which is very specific for that particular gene. It is not having any non-specific binding with any other region in the genome. That particular sequence, you call it as a probe, and you fluorescently label them. Remember, the fluorescent labeling is done not by you. It is done by different companies. So you have to send that particular sequence to a company. They will make that particular nucleotide sequence as a probe for you, right? The probe making is an integral part of the so-called probe-based quantitative real-time PCR, right? So the different types of probes are used in real-time PCR. The Tachman probe is the most popular one, much similar to what we talked about, cybergreen, right? And we have other kind of probes, molecular beacons, scorpion primers, and hybridization probes. These are some of the other options what you have, right? Let's see, now the pictorial I will explain. That would help you to understand in much better manner. For example, it's a Tachman probe. So what you have in the PCR cocktail now? in the previous cyber green based, you have everything which is a normal complement or normal composition of your PCR. You have extra ingredient, that is a dye. Here, you have the same option here. You have everything plus one extra. That one extra here is probe, which is fluorescently labeled. I hope you understand this. So one is, in the previous one, you added a dye. The dye is cyber green or the variants I already told you. In this particular case, instead of a dye, you are going to add a fluorescently labeled probe, right? So now that is put here pictorially here. And you see two letters here. One is R, another one is Q, right? Here comes a slight finer details. This particular probe or a stretch of nucleotide which is complementary to your target is labeled with two different chemicals, right? One on 5' end, another one on 3' end. One is called as R, another one is called as Q, right? R stands for reporter, Q stands for quencher, right? And this setup is important in real-time PCR. Let's take this lecture forward. And what happens is, when you start setting up the PCR, what happens? When intact, the fluorescent dye of the reporter is quenched due to its proximity to the quencher. What happens is that the cycle 1, you have everything plus the probe. The probe is having two molecules attached to it, one on 5' end, another one on 3' end, right? One is a reporter, that is technically called as fluoroprobe, and another one is technically called as quencher. Quenching means it is suppressing the fluorescence which otherwise would have come from the fluoroprobes, right? It is something like, as long as the quencher molecule is there, the fluoroprobe cannot emit the signal, right? In other terms, in the presence of quencher, the fluoroprobe is unable to emit any fluorescent signals, right? Now, if you want to see the fluorescent signal coming from this particular setup, what is needed is the quencher need to be removed. The moment the quencher is removed from this probe, the fluorescent molecule will give you the signal, right? This is the setup in probe-based real-time PCR. What happens here is, please go through it. When intact, the fluorescent dye of the reporter is quenched due to its proximity to the quencher. The chemistry here is, as long as the quencher is in close proximity of your reporter, also called as fluoroprobe, the ability of the fluoroprobe to emit the fluorescent signal is switched off, right, or quenched, right? The probe hybridizes to the target because it has got as complementarity and specificity, because you only designed that particular probe. It has got a very specific complementarity towards the target. Remember, I'm not talking about primer yet. I'm only talking about the probe. Now what happens? You have a polymerase enzyme which is going to add the nucleotides, right? So it is adding the nucleotide in 3' to 5', but the same polymerase enzyme is having additional activity, what is called as 5' to 3' exonuclease activity. In fact, we are exploiting this particular property of this polymerase enzyme to remove the quencher which is in close proximity to the fluoroprobe, right? So what happens when the primer is getting extended with the help of polymerase enzyme? The same polymerase enzyme will encounter the probe which is bound to the target somewhere away from the primer binding site. So it started cleaving whatever nucleotide coming in its path, including the quencher and also the fluoroprobe, right? So it is chewing off the nucleotides from 5' to 3' exonuclease activity of this TAC enzyme, which cleaves off the reporter, not only the reporter, but also the nucleotide sequence and also the quencher. So now the quencher and fluoroprobe, they are separated from each other. right? And the dye is released into the PCR cocktail, right? So there is no DNA binding here. The dye is freed from the quencher. When the dye is freed from the quencher, technically it can emit the fluorescence, right? So now the reporter is separated from the quencher and the fluorescent signal started appearing, and the signal is proportional to the amount of amplified product in the sample. The next slide will give you much better manner. This is the fluoroprobe, much similar to the primer. You have the fluoroprobe, that is the reporter. You have the quencher, that is written as Q here. And what happens in the cycle 1? You have a double-stranded DNA template. You are subjecting the template to 94 to 96 or 98 degrees Celsius. That is what is called as a denaturation temperature. What happens? It is made into 2 single-stranded DNA, right? So the moment 2 single-stranded DNA is formed, what happens? In a mixture, you have DNTPs, you have primers, you have magnesium, you have all other ingredients which is needed for the reaction to happen. So the primers and probes will go and bind to the complementary sequences on the target. So I'm just depicting the probe here. Primer will come a little later. So the probe, which is synthesized or custom made by you with the help of some companies, they added the quencher, they added the fluorescent fluoroprobe on the nucleotide sequence, that you call it as a probe, and that is binding to the single-stranded DNA here, right? Now what happens? So now this is already bound to the single-stranded DNA, and it is happening parallelly. So you have the primer somewhere is going to bind to another region in the target, which is having the complementarity, right? And this primer bounds, and it start, priming the reaction with the help of TAC enzyme, right? So what happens now? So now the primer start extending it. So the green color one is the TAC enzyme. It start extending the primer. So once it start adding the nucleotide, whatever comes in its way, that is in the form of probes, right? It start ripping off. For example, here, first, what do you call, the quencher is removed, and what do you call, the other nucleotides in between, and also the so-called probe, everything is removed now. You can see here, only the primer is here. The primer binding on the complementary template, the DNA polymerase. Now what happens? This is the final setup. So the specific probe, which the 5' to 3' exonuclease activity of the polymerase removes the fluoroprobe, probe nucleotides on the quencher, and the fluoroprobe emits the fluorescence after detachment from the quencher, right? So what happens here is we are using 2 sets of the complementary sequences. One you call it as a primer, another one you label it as a probe, that is deployed in the probe-based real-time PCR technique. So let's see what happens here. Once the amplification is completed, in the PCR mixture, you start seeing the fluoroprobe and also the quencher floating in that. Once the fluoroprobe is away from the quencher, it starts giving the fluorescent signal, right? So this is what finally happens. So this is quite different from the cyber green one. In cyber green, the bound dye is detected, right? But in the case of a probe-based one, the unbound dye is detected. That's a fundamental difference between these 2 techniques, right? So there is an accumulation of free fluoroprobe in the PCR mixture with every passing cycle that add the net fluorescence coming from the PCR reaction mixture, right? That is completely different from the previous one what we talked, that is cyber green based detection, right? The very important point here is 5' to 3' exonuclease activity of polymerase, which removes the probe nucleotide and also the other things which is already there, right? Now I'll take 2 slides just to explain how the quencher is doing the quenching function. So 2 mechanisms are is operating here. I think you need to read more on this, FRET and non-FRET. FRET, different acronyms. The FRET is an acronym, but different expansions are there in the literature. These 3 are the most common one. Forster resonance energy transfer, also called as fluorescence resonance energy transfer. It is also called as resonance energy transfer or electric energy transfer. All are meaning the same. So in the FRET quenching, there are three important points: the reporter and quencher. They interact through the space between these two, and not very temperature and solvent dependent. And the absorption spectrum is unchanged, whether it is bound state or unbound state. That is some of the traits of this FRET quenching. And if you want to, and there is another kind of quenching that is a non-FRET quenching. Here, the physical contact between the reporter and quencher is here, and the temperature and solvent dependent, and the absorption spectrum of the reporter is distorted. So these two are explained basically two are highlighted to explain how the quenching is happening on the fluoroprobe with the help of quencher, right? If you want to know more about this FRET and non-FRET quenching, please this particular website, the quenching mechanisms in nucleotide probes. It's a beautifully designed, student-friendly website. You can just go through this website and every small finer details are explained in a much better way. So please remember BioSearch Technologies. It's for the benefit of students. I brought this slide, and all finer details, how this FRET and how this non-FRET quenching is really happening with all basic the physics details is given in this particular website. Little more on the type of reporter, the fluoroprobe. FARM, JO, and TET, they are very popular. TET stands for tetrachlorofluorescein. FAME stands for fluorescein amidite. And JO is 4,5-dichloro-2,7-dimethoxy carboxyfluorescein. These are some of the examples for the fluoroprobe, what is being used in probe-based real-time PCR. And this is some examples of quenchers. And we have BHQ, that is black hole quencher 1. We have TAMRA, that is a tetramethyl rhodamine. And MGP, that is minor groove binder. These are some of the examples of quencher. Remember, the probe is bound with the two different molecules. One is the fluoroprobe, another one is the quencher. And again, the great deal of discussions and information is already available. Which two combination we have to go, right? Because I've already put here 3 different dyes and 3 different quenches, and people use the 2 different combinations. The time is really not allowing us to explain in depth. You can please enrich your knowledge by visiting those websites. The advantage of the probe-based real-time PCR, it's very, very specific because you are deploying 2 different probes. One you call it as a primer, a conventional term for real PCR, and another one is a probe, which is custom made by adding 2 different chemicals. One is a fluoroprobe and one is quencher. And multiplexing is possible because you change the fluoroprobe, which is having a different excitation or different emission spectrum. This is how the different color systems in PCR, real-time PCR works, right? If you go to market, people will ask whether you want a 4-color system or 5-color system. Basically what they mean is the ability of the machines to capture light with the different kind of wavelengths. So they mean only that, right? And a disadvantage, definitely you need to custom made your probes. That is a technically demanding, and it's a pretty expensive. And also there are reports that false results sometimes it appears. And this is the application a little bit. I'll see what you call one of the applications in real-time PCR is what is called as reverse transcriptase PCR. I think all of you must have studied this, especially the viruses with RNA as a genetic material. This is one of the tools normally people use, right? The reverse transcriptase, I just brought this slide for those people who is not having a great idea about it. Remember, RNA is converted into a cDNA, and the cDNA is used as a template here. This technology is what is called as a reverse transcriptase PCR, right? That is slightly different from real-time PCR. Of course, in real-time PCR also you can use cDNA as a template to study the expression or to study the copy number of the target. So we'll come to that a little later. So 1-step and 2-step PCR, I think many of you must be knowing. So in 1-step real-time PCR, every reaction is carried out in a single tube. So I already told there is a conventional PCR which is added with a fluorescent dye. It becomes a dye-based fluorescent dye-based real-time PCR. If you add a fluorescent probe, then it becomes a kind of probe-based real-time PCR. In the whole cocktail, if you add a reverse transcriptase enzyme, then it becomes a one-step real-time PCR for RNA templates, right? So this is one step. And in two-step, there are situations where you need to go for two steps also. In two steps, what you do, you synthesize or you make enough of cDNA and you preserve them somewhere in the lab and use them on the need-based in a second application, that is a real-time PCR. Here it is two steps real-time PCR. So it's very simple. Here everything mixed together in a single tube, that becomes a single one-step qPCR, and you segregate that into two independent work. You make a cDNA and preserve them and use them as a template in the real-time PCR, then it becomes a two-step real-time PCR. Requirements, of course, you need a machine and a template, that is the target, whatever you want to study about, and of course primers. So I just highlighted one particular website here that really guides you to synthesize the real-time PCR primers. So you just Primer Quest is a tool that is integrated in a company website, what is called IDT, that is Integrated DNA Technologies. And this is one of the most popular choice for designing real-time PCR primers. And if you have time, you can get into this particular tool. Primer Quest is a tool, right? And you have Tachman Probe, in the case of probe-based, or you can use the dye cybergreen in the case of dye-based. Of course, you need to have DNTPs, you have magnesium, you have, of course, the enzyme, which is a normal ingredient of your PCR. Of course, the buffer, it varies from system to system. This is a website of the Primer Quest. Excuse me. So you can register your name, generate your own username and password, and you can use that particular portal for your routine kind of work. Any kind of primer designing, you can use this particular website. Cycling conditions, there is not big difference. You have the conventional PCR cycling conditions. You have a initial denaturation, you have annealing, extension, and of course the repeat. Usually we go for around 35 to 40 cycles, and of late we are restricting up to 30 cycles. So that is one of the options nowadays. Excuse me. So up to this point, what I conveyed to you is some of the fundamentals of real-time PCR. The focal point is on dye-based detection and also probe-based detection. From here on, we'll just move on to a little more on the visualization of the so-called PCR reaction, right, and the different stages. Remember, the typical real-time PCR reaction, you can divide into 4 steps. Step number 1, what is called as linear ground phase. Step number 2 is early exponential phase. And third one is linear exponential phase, also called as a log phase. And last one is the plateau. This is common across all real-time PCR experiments, whether it's a dye-based or it's a probe-based, whatever. This is a linear ground phase. What happens here? In the first phase, the PCR is just starting. The fluorescent signal has not risen above the background. Remember, at the start of your reaction, the unbound dye is still there, right? The probes with quenching is also there in the case of probe-based, right? So that is at the ground level. So this particular phase is somewhere here. Just reaction progress now into the cycling conditions. That is called early exponential phase. The second phase is where the fluorescent signal just raised significantly above the background. The cycle at which occurs is called cyclic threshold. This is so critical in real-time PCR. This is also called CT or CQ. I think you might have encountered this acronym, CT values and CQ values. So this is the early exponential phase. So that means the intensity of fluorescent signal started visible in the form of a rising curve in the PCR machine. Linear exponential phase is where the fluorescent signal is doubling in every passing cycle, that is quite visible in the form of linear increase in the curve, this phase, right? This is also called log phase. In the linear exponential phase, PCR is in its optimal amplification stage with doubling PCR product in every cycle. And the plateau where everything got exhausted, there is no more DNTPs, no more enzymes, no more dye, because everything is a limiting factor here. Everything got exhausted. Then even if you continue the cycling and the curve is no longer raising, it takes a plateau. This is a typical 4 stages of real-time PCR. And come to the data analysis. Baseline of amplification plot is one of the popular jargon we use in real-time PCR. That's the initial cycles of PCR. There is very little change in the fluorescent signal because the machine is unable to see the fluorescent signal coming, or the capability of the spectrophlorometer to capture the signal is not sufficient to display in the form of a curve, right? That is what is baseline amplification plot. And increase in the fluorescence above the baseline, which indicates there is an accumulated PCR product. So that particular junction is called CT value or CQ values, right? If you have a small CQ value and higher CQ value, can anyone tell the CQ value of 10 if I'm getting for a target, what it really implies? Template concentration is very high. If CQ value comes in the late, what does it implies? The template is very, very low. And try to remember this point, okay? Yeah, threshold line, a point at which the reaction reaches the fluorescent intensity above the background, which I already told you. And it is set in the exponential phase of the amplification for most accurate reading. Cyclic threshold or CT, or sometimes it is called CQ also, the cycle at which the sample reaches the threshold level. And CT value of 40 or more is usually considered as failed reaction in real-time PCR. Just move on. What is melting curve? This is another point normally people do. Melting curve is so important, especially in the case of dye-based real-time PCR, just to know whether the target amplification is really the target and it is not the non-target. So how do you rule out that? You need to perform melt curve analysis. Why it is so important in the case of dye-based fluorescent real-time PCR? Anybody? Because cybergreen is non-specific. So necessarily you have to do this melt curve analysis just to tell the readers of your paper or your work that the primer is very specific. So you need to do melt curve analysis. So what's happening here? You have a double-stranded DNA, the dye is bound in the minor groove, right? So we know that when it becomes a single-stranded DNA, the dye is going to come out of the double-stranded DNA, right? So when the double-stranded DNA will become single-stranded DNA, you have to raise the temperature. You have to melt the double-stranded DNA into 2 single-stranded DNA. That's what we do that for. That purpose only it is called melt curve analysis. So what happens? So as the temperature is increasing, so when a particular temperature is reached at which your double-stranded DNA become 2 single-stranded DNA, then there is a sudden drop of your fluorescent signal. So this is what is called Mel curve analysis. So if there is a many off targets or non-target amplification, how the curve looks here, anybody? You have to interpret the results also, right? So in a typical, a single target priming, you get a curve something like this. You remember this particular picture, right? There is only one peak. If there are multiple targets, multiple amplicon, the primer is a poor primer, it amplified multiple targets, then what happens? You see more number of peaks. So you can accurately assess how many non-targets got amplified using the bad primer what you used, right? This is very important, right? So Mel curve, changes in the fluorescence when the double-stranded DNA incorporated dye molecule melt into single stranded as the temperature is raised. When double-stranded DNA bound cybergreen dye is heated, a sudden decrease in the fluorescence is detected at Tm, that is a melting temperature. So it is one of the mandatory steps whenever you are doing a dye-based real-time PCR. Right? So again, the changes in fluorescence and the changes in temperature is plotted against the temperature to obtain the clear view of melting dynamics. That's what it has happened here. And this is also performed by the machine itself, nothing to worry. Nowadays, the software is integrated in the real-time PCR machines. It gives you all the data of your experiment. Now, I'll take you to a few slides which highlights some of the potential applications of this real-time PCR. The number one is quantitation of gene expression by real-time PCR. There are two analysis is possible. One is absolute quantitation of the target, number one, and number two is the relative quantification. These two are possible in real-time PCR. The level of expressed gene may be measured by absolute or relative quantitation, quantitative real-time PCR. The absolute quantitation relates the PCR signal to the input copy number using a calibration curve, while the relative quantitation measures the relative changes in the mRNA expression level. Let us take a little more on this. Two broad applications of this real-time PCR is just highlighted here. One is absolute quantitation and another is relative quantitation. In absolute quantitation, which determines the expression level in absolute number of copies, and in relative quantitation, which determines the fold changes in the expression between two samples. I'll try to explain in one particular slide in future. Little more on this absolute quantitation. What is needed here is you need to have a standard curve. So normally what you do when you want to quantitate something, whether it is spectrophotometer based quantitation, normally we do a standard curve making with very high, what they call closer to 1, the regression values, right? One is absolute value. We try to achieve that, the regression coefficient. Same thing here. You need to make a standard curve using a known quantity of your target. So that's number one. Number two, the standard curve is based on a serial dilution of the sample with a known copy number, and the CT value or CK value of the each standard is plotted against a logarithmic of the known concentration, and the standard curve is then used to estimate the concentration of the unknown samples, right? This is nothing but the routine kind of thing what you do in a spectrophotometer. There is no difference. In the case of relative quantitation, you are comparing your target with something else which is constantly expressed in the plant, right? Suppose I'll take one small example here. In plant, when it is infected by viruses, there are a number of genes got differentially expressed. Remember, there are genes in the plant, what is called as housekeeping genes, sometimes whose expression is not changed at all. It is constant, whether the plant is infected or not, whether the plant is subject to the differential condition or not. Those genes are what is called as reference genes, right? And you compare the expression of your target genes to the reference gene. So here comes the relative quantity. In relation to a normal expression of a housekeeping genes, how the target genes which you feel is vulnerable for viral infection and how those genes are behaving in the plant. So this is what is called relative quantitation. Changes in the gene expression in a given sample relative to another reference usually the reference is an untreated control sample or analyzed, and calculation methods are for relative quantitation is two different methods. One is standard curve method, another one is comparative CQ method or comparative CT methods. And some terminologies we use, normalization and housekeeping. Normalization, that is the help of some endogenous control, which just now I told. There are genes whose expression is not at all altered, whether the plant is infected or not, and those genes are considered as the reference genes in real-time PCR. This is absolute quantitation, which I already told. There is a standard curve, and you compare your samples with a standard curve and try to decode how many copies of your target is there in the sample. And nowadays, there is a kind of website which helps you to accurately quantitate how many copy numbers are there. You don't have to do any calculations. This is one such website, the scienceprimer.com. So scienceprimer.com is a website where you need to give two input values. One is the amount of amplicon, what you decoded from your real-time PCR curve, and second one is the length of the double-stranded DNA amplicon. Only these two values you need to supplement here in this formula, and that is going to give you the copy number of your targets, right? So you don't have to really do the calculations in your notebook. You just click this website. That takes you to a window something like this, and you supplement, you just feed those values, whatever you decoded from your CQ standard curves, your amplification curves, and those values, you just click of your button, you start getting the number of copies of your samples. So this is very simple. So what is needed here is you need to X and N, you have to supplement in this particular table, in this formula, if you want to do it manually. Otherwise, you just feed the values here. So one is the amount of DNA in terms of nanograms, and also the length of your target. So that is going to give you how many copies are there in the sample, right? This is the softwares are already there, and it is publicly available. You can use directly in the online tool. Yeah, some consideration for absolute quantitation. It's quite easy because you need to have a standard curve that is very easy to make, and of course you can easily decode the number of copies from the standard curve. Relative quantitation is, which I already told, in relation to a normally expressed gene. Popularly, those genes are called housekeeping genes. However, the gene which is vulnerable for viral infection is behaving, either it's upregulation or downregulation, it can be related to those genes. So maybe in one slide I will explain that. In any relative quantitation or real-time PCR for gene expression analysis, you always remember this particular checkerboard. So you need to have 4 inputs here. One is called as calibrator, another one is called test, and that is row-wise. In column-wise, you have a target and also reference. I'll just explain what this each quarter is meaning to us. Calibrator is something what is called as a mock plant, or the plant which is not inoculated with the so-called virus or pathogen. Is it clear to you? This is called calibrator. The test is the one which is inoculated with the pathogen or the virus, right? And target is the gene which you are studying or the very objective of your experiment. For example, if you want to study a particular gene which is having role in host resistance, that is a target, right? You want to study the behavior of that particular gene expression in relation to what is called as a reference. The reference is housekeeping genes. So whenever you do gene expression analysis using qPCR, you first make this particular checkerboard, right? And you define what is a calibrator here, and what is a test here, what is a target here, and what is a reference here. Remember, the reference gene amplification need to be standardized. You cannot go with the literature, whatever is there. You need to standardize and ensure that the quantum of target, what is amplified, is constant across different experimental conditions. So that is one of the criteria of the so-called selection of reference genes. Sometimes you find even a dedicated paper on this, just to standardize what can be the right reference gene for qPCR, RT real-time PCR for gene expression analysis. People do it deliberately. They don't go with the literature reports. What can be the right reference gene for gene expression analysis? You need to really standardize that. Okay, so here, for example, you have a calibrator, a mock plant. You have a test, the same mock plant. Remember, the mock plant and test plant, they should be genetically identical. You cannot take a completely different species level boundary. You have to go for exactly the same genotype, right? And target, of course, that is your primary objective of your experiment. And reference, you need to standardize, right? Now, once, for example, I will tell you the test target. What can be the target? It can be in the typical plant pathology experiment, it can be effector genes. Right? You want to study how the effector genes get expressed in the presence of the host material. That is one option. Put the other way around, it can be the receptor genes which is there in the host, how it behaves in the presence of the pathogen. So these two can be studied using this approach, right? And the most common statistical approaches are, you remember this, I'm not going to elaborate that. Very popular methods are three popular methods. Levac method or double delta C method is a very most popular one. We have delta C method using a reference gene, and also FAFL method. These three methods are very popular in the gene expression analysis, right? And for relative quantitation, of course, one of the biggest advantage of relative quantitation, you don't need to know the targets, right? The quantum of targets. With that, I'll move on to some of the applications. We are already explained in length. A gene expression analysis is possible. You can detect the mutant genes. You can copy number estimation. The potential application in pathology is pathogen and microbe detection or quantitation, or even viral load estimation is possible with the help of absolute quantitation using standard curve. And of course, SNP analysis is also possible, and many more applications which you can design in real-time PCR. So these are all some of the machines, what is very popular nowadays. And light cycler 96 from Roche is one of the most popular one. We have from ABI, number of machines are there. From Bayreuth, these are some of the companies who sell these kind of products. So with that, I conclude. I think we had some interruption in the form of audio quality that delayed this presentation by another 20 minutes. So otherwise, I hope everything went on well. If you have any quick questions, we'll take it.